What is? Cool. Yeah, and I texted the link to you, so if it's just like Sean wanted, I didn't realize that. But.
Right. What is the practice models now? Are people a lot of so it's mostly consolidating, right? I guess that's
You should wave.
right. Yeah. I guess the small ones can still work. Yeah, right. Right. No, it doesn't. Yeah, my wife's a psychiatrist. She's out of the because then she's like 50 patients a day. She's psychiatrist. Actually, that really actually figure out what's going on, and also talking to her. So she spends hours talking to insurance. So it works for that. Psychiatrist. Well, the patient yeah, they come in 5 minutes. How you feeling? Better? Okay. Not better. Yeah.
That was his phone vibrating. Could you hear it? Or?
Oh, my phone just vibrated.
Yeah,
His, I think Joel texting both of us.
And that's fine. Do you want to
Check in on screen. Link to the left stream.
because EMR and then all facility fees and whatever. Okay, so you can do a lot of that.
Damn.
My wife does that kind of thing because she has patients who do really well. And they email her like, oh, my dog got lost and blah, blah, blah. And they'll email her, we found the dog, like, Yeah, really, absolutely. Especially as a psychiatrist. The whole thing is basically that's right. Yeah. How are you doing, Barbara?
All right.
Yeah.
All right, yeah.
I haven't seen you since
Yes.
which is good. Yeah.
I'm fine.
Yes, I told everything is going fine.
I said without meeting him in advance, you have to directly.
You're not actually being by the day?
Yeah, it's growing. It's been a lot of fun. Yeah, Ben started on the ground floor, so when he came, we were much
Oh,
smaller.
yeah.
Yeah, and we're doing a lot of mics. And I also use research on my lab, but I use other collaborators on Stephen Alzheimer. What do I? Oh, the subject now, right. We have to decide them. Yeah, no, it's interesting. Even for mice, it's very, you have all these rules and regulations.
Okay.
What flying, no one cares about. So. Oh, yeah, no one
Oh,
cares.
they don't have enough lobby.
No one cares. That's right. No one cares about
lobby.
Stop.
No one likes. No one really likes the mouse either, but But somehow that's not the
It's just Stan.
Ben's an expert on mouse behavior now, so we have a station on mouse, some mice in my basement that gives some advice. How to think like a mouse. How to think like a mouse.
And when they're gonna be awake, and when you should go out.
Exactly. Did you try this morning?
I go, and I'll stay overnight.
One night.
Super quick jump. Remember I was asking about the RNA sequencing project? Like a while ago. That so that's what Josh did for us. So I just said I'd introduce you guys. In case he comes up with stuff in the future questions.
You came that night? You came after I left. Yeah. Oh, that's right. That's right. I talked to her.
Good luck.
Try not to swing.
Yeah.
It'll be a fun hour.
Hopefully Justin has a class. That's his later.
I can feel it. I could be cool.
Huh?
No, she's actually we're talking about less and less of this, but it actually works. A lot of the things are actually
Is that silent?
so that's why it works. It's like your dentist. Balance, yeah. So it's actually not like you don't have, it's like you pay, and then you start. You pay, you have no overhead. It's a great model for this type of practice. No problem.
In terms of sequence-wise,
You're
No, mine. I mean, no one from my house, only my friend. The ones who are willing. Oh, that's great.
Oh wow. Yeah, which is he told me
Did he make you like
because everybody gets paid for talking about
it was funny
Again, I just Skype on. I test.
Yeah, good.
told her she's drawing on like the first time I've done something like that. First time all these people that took over before
My dad,
Now she's swearing because maybe not.
it's very funny.
Quite formal. And also, no, I couldn't come. And also, I couldn't.
It's more empty on more active.
I almost never had any teacher in my
Me losing everyone else.
Oh, this is but like, they're not going to do it. So it's going to be like 4 feet away. So it's going up, or down, but either way. It's like, and also it's probably more fun like that. I doubt they It looks like it comes up. But I mean, like, you gotta lean on that.
All right, why don't we go ahead and get started? So good afternoon, everyone. I'm Mark Liu, and it's my tremendous honor to introduce Ben Bell for his thesis defense talk. So when I thought about Ben and his time in the lab, 2 words that start with the letter C came to my mind: courage and commitment. So as many of you know, Ben is a pretty courageous kind of guy. I remember when he rotated in the lab, Ben lived in a, let's say, interesting part of Baltimore. And one day someone stopped him and he handed his wallet. And Ben said, "No, I don't think so." And he walked off in a manly sort of way. All right, okay, so maybe he ran away. But the point is that I thought that was a rather gutsy thing of Ben to do. Ben is also really brave and courageous in his science. So not only was Ben the first graduate student to join the lab, when he joined our lab, the lab was new, just newly established, and didn't really have much of a track record. In addition, Ben decided to take on a mouse project in the lab that only worked with fruit flies. So I thought that was also pretty gutsy. When I stopped to think about it, it's really incredible all the things that Ben has done. In addition to all of his experiments for his project, Ben also single-handedly set up all the mouse equipment, all the mouse protocols. He built the mouse behavior room. He maintained the mouse colony. Our lab really owes a tremendous debt of gratitude for Ben for doing all those things. Because Ben had to do all this mouse setup, initially in the early days, his project moved a little bit more slowly than we would have liked. But then over time, he started to really hit his stride and his project was really picking up steam. And then, as many of you know, May of 2017, Ben was involved in a serious hit-and-run accident. That night was probably the worst night of Ben's life, but I would argue that it took even more courage to make it to the grueling rehab that it took to restore him back to full health. At the time of his accident, Ben had made significant progress on his project, but he had not yet made many of the most important discoveries that he'll be telling you about today. And yet, he never wavered in his commitment to not only finish a PhD project, but actually see this thing truly through and truly understand the fundamental function of the gene and circuit that he was studying. So in closing, I'm tremendously proud of all that Ben has accomplished. As we'll hear about today, I truly believe that the beautiful and important work that Ben has done will change the way scientists fundamentally view arousal. You know, when graduate students join our lab, they come to us as trainees, full of energy and enthusiasm, but little in the way of research experience. Now, after years of training, I can honestly say that I learn more from Ben than he learns from me. And so, as Ben now prepares to leave my lab, another C word describes him, and that is my colleague. Ben?
So, thank you, Mark. That was really a very nice introduction. I'm very appreciative. Can everybody hear me? I'm going to move around a little bit so I'm not always by the mic. You can still hear me. Good. So thank you everybody for coming today. During our time, I'm going to talk about the main project that I've been working on while I've been in Mark's lab working on my PhD. When I set out on this project, we were examining a novel protein that we had predicted was involved in regulating sleep in mammals. But over the years, we've shifted our focus a bit, and we realized that the circuit of interest actually broadly affects arousal in mammals. So in humans, arousal comes in peaks and valleys. It rises sharply when we first wake up, stays high in the morning, with then a little bit of a dip before it comes back up for the rest of the day. Welcome, everybody, to the afternoon song. 2 p.m., time perfect. The best time for me to study my thesis, but I'm going to try and prevent people from going down, too far into that slump by keeping everybody interested with what I think, anyway, is a really compelling story. So here's just a brief outline for what I'm going to talk about today. I'm going to start with some background on the circadian system, how this internal timekeeper regulates a vast swath of physiologies. And then the first story I'm going to talk about is this undescribed protein called the M-weight that the lab first discovered in fruit flies, and I've shown that it acts similarly in mammals, regulating arousal under control of the circadian clock. Now, when I first joined the lab, part one was supposed to be my PhD, but things never really stayed static like that. And so in part two, I'm going to tell you about a sort of a canter to the left that this project took. When we found that the neuronal circuit that expresses M-weight comprises a network that's fundamental to arousal in these mice. Because I know I tend to run long and I don't really want to keep people here all day, I'm going to limit my future directions to the end if there's time, but I will have to be if there isn't time. But first, let's talk circadian. I know I literally just said that I tend to run long, so I'm sorry for this, but our story starts back about 3.5 billion years ago at the very beginning. As long as there's been life on this planet, organisms have had to organize their biological processes around the consistent changes in the environment over the course of 24 hours as the Earth revolves on its axis. Just about every living creature is subject to these changes, and the animals that could predict these consistent changes and adapt to them were then going to be evolutionarily favored. If we fast forward to the present, just about every organism aligns their biological processes to these rhythmic changes in their environment. These processes are then called circadian rhythms, defined by, and in the period of around about a day, and they take about 24 hours to go all the way through. There's a remarkable diversity of these rhythms throughout the kingdoms of life, from bacterial metabolism to animal feeding, to plants that open their leaves during the day and close them at night. A huge amount of life aligns to the rhythm of the Earth's rotation. But it wasn't until about 1729 that a French scientist named Desmarais conducted an experiment that's really kind of brilliant in its simplicity. He asked a chicken and egg question, right? He said, were the plants opening their leaves in response to the sun or in anticipation of it? So he took these same plants and he put them in an all dark room away from the sun's influence. And what he saw was that they continued to open their leaves during the daytime and close them at night, even without the sun's influence. The plant had its own internal timekeeper. Exactly what this timekeeper is, though, has eluded scientists from the 1729 experiment all the way up into the 1980s. Only last year, the Nobel Prize in Physiology and Medicine was awarded to these three scientists, Michael Rosbash, Jeffrey Hall, and Michael Young, who in the 1980s began to tease apart the genes and proteins that actually form the gears and the machinations of this clock using forward mutagenesis screens in Drosophila. The mechanism they discovered is called a transcriptional translational feedback. And in brief, it's formed when clock and BMAL transcription factors drive expression of period and cryptochrome here. These genes are then translated into proteins in the cytoplasm, where they dimerize, and then translocate back into the nucleus, where they inhibit their own expression. Working out this really nice structure of how Drosophila keep time isn't why Rosbash, Hall, and Young won the Nobel. What they really showed was that there's a commonality between both the circadian clock in flies and the circadian clock in mammals, and lots of other animals in between. Although the individual protein gears of this clock can vary somewhat from animal to animal, it's this overall structure that remains remarkably similar. Over this 24-hour period, this clock can then regulate a vast network of genes and physiological processes and behaviors and outputs. In fact, our current best estimate is that over 50% of the transcriptome is clock regulated, and a number of physiological processes as well. For example, we know that in humans, hormone levels rise and fall over the course of the day, like testosterone that peaks up here in the morning. We know our blood pressure and our body temperature are highest in the evening and then lowest at night before rising again to wake us up in the morning. And we know that all of these rhythms here work in concert and in sync with each other each and every day. But the circadian output that I think we all are most innately familiar with is the sleep-wake cycle, right? The sleep-wake cycle has to be the most obvious and protracted 24-hour rhythm that most animals experience. We all don't exactly know when we start secreting melatonin in the afternoon, but we're all bona fide experts in knowing when we're asleep and when we're awake. So looking at sleep, sleep is a relatively well-defined behavior. It's shared by just about all animals with enough neurons in one place to call a brain. Some of the main attributes of sleep we can define behaviorally, right? We know an animal that's sleeping is going to have sustained quiescence. It's not going to be moving around, not adjusting itself for a while, and it will have an increased arousal threshold, right? It won't be as responsive to external stimuli. But if the stimulus is strong enough, if you shake the person next to you, they'll hopefully jolt out of wakefulness, jolt out of sleep. And that's because sleep is quickly reversible, and that's what distinguishes it from coma or hibernation. Sleep also has a number of physiological characteristics. For example, our brain activity during sleep is different than it is during wakefulness, and we can measure that using electroencephalography, or EEG. And I'll talk more about that in a little bit. It's also important to know that sleep is regulated by two main processes. There's the sleep homeostat, which monitors and produces sleep drive, and the circadian clock regulates the timing of sleep, the onset and end offset of sleep. So the underlying purpose of sleep, the original underlying purpose of sleep, is still hotly debated in the field and the literature. There's actually just a paper that came out on Tuesday that has a new idea for it. But we don't really know exactly why we have to sleep. What we do know, though, is that if we don't get enough sleep, or in the case of shift work, if our sleep is mistimed from our circadian clock, there are a number of damaging health consequences. For example, long-term sleep deprivation can lead to a risk of type 2 diabetes, it can lead to an increased risk of heart diseases or cancer. It can suppress your immune function. These are all long-term consequences. On a more acute time scale, this is a plot that I personally am a lot more intimately familiar with. Here, human performance on the y-axis drops precipitously over amount of hours spent in wakefulness. And you can see that these are actually this is just a sort of a long day in lab, right? 18 hours, you're already operating below the level where you need to be. In fact, researchers in the UK have shown that trying to drive after only 18 hours a week is approximately equivalent to blowing a 0.08 blood alcohol on a breathalyzer in terms of reaction times and other measures. So we know that sleep is important, and we know that the circadian clock times sleep. But for all that we as scientists know about these two features, we don't actually know the mechanism by which the clock can affect sleep behavior. So to address this gap, before I joined, the lab turned back to large-scale mutagenesis screens in Drosophila, and they were looking for genes which impacted circadian regulation of sleep. So after screening many thousands of fly lines, they found a single fly mutant with significantly disrupted sleep. As you can see here, on the y-axis is sleep amount, on the x-axis is 24 hours. A wild-type fly in gray will do most of its sleeping at night with a nice little daytime siesta, a daytime nap. This mutant, shown here in black, has significantly reduced overall nighttime sleep, as well as this increase in sleep latency. Sleep latency is the time from when the lights go out to when the fly first gets to sleep. And the fact that this was increased here suggested a deficit in sleep timing. So in the grand tradition of naming genes after their fly mutant phenotype, the lab called this gene wide awake, and set out to understand its role in sleep timing. I really don't have time to go through all of the data from the fly awake story, so please trust me that it is beautiful, it's really nice, I recommend that you read it. But here's the model that the lab projected for this wide awake protein in flies. Wide awake is expressed in arousal promoting clock neurons, which fire regularly during the day to drive arousal and drive wakefulness in the fly. As the day progresses and the circadian clock ticks over, it drives an increase in expression in this wide awake gene. Wide awake then causes an upregulation, an increase in the receptor, the GABA-A receptor, which is sensitive to this inhibitory neurotransmitter GABA. This increase in inhibitory tone quiets the arousal promoting cells, allowing the fly to get to sleep in a circadian appropriate time. Then, over the course of the night, protein degradation breaks down the wake proteins, and the wake breaks down the GABA-A receptors, and the whole cycle can start afresh in the morning. And as I said before, the circadian clock and the sleep and arousal dynamic are evolutionarily very ancient. So we anticipate that a lot of these systems are going to be shared throughout many different stages of animal life. So the lab looked for mammalian homologs of this wide awake gene, and they found a single hit. Now this hit, ANKFN1, is a virtually undescribed protein, and it's the single mammalian homolog of wide awake as measured by sequence similarity and sequence identity seen here. ANKFN1 is also the only gene in the mammalian proteome that has this specific series of protein domains. But most strikingly, ANKFN1, or what I'm now going to be calling M-wake, the mammalian version of wake, is strongly expressed here, the suprachiasmatic nucleus, the SCN. This region is the core circadian clock center of the mammalian brain. And it's similar to those clock neurons that we saw in flies. So all of this was far too tempting and too much of a coincidence for a circadian lab to pass up. And so in, I think it was 2013, I joined their fly lab to do mouse research. All right, so the first part of the story, we wanted to understand, we used our understanding of the fly model to test our hypothesis that M-wake in mammals is the functional homolog of wide awake in flies. And as I mentioned before, we were originally encouraged by the fact that M-wake is expressed in the SCN. The SCN is a master circadian pacemaker of the mammalian brain. It's a small cluster of neurons in the ventral hypothalamus. It receives direct photic input from intrinsically photosensitive retinal ganglion cells in the eye. Those light cues then set the timing of the endogenous clock here in the SCN. And the SCN then sends signals to output networks as well as synchronization signals to other cells and other tissues that have their own internal timekeeping. So the fact that we saw M-wave here in these core clock neurons highly suggested that it was involved in circadian timing of the mouse. And so to understand exactly what its role is, we had to develop two new mouse models. To make our mouse models, we use CRISPR-Cas9 genome editing. I know that for most of the people in this audience, I think CRISPR-Cas9 is a pretty old hat. And for other people, you've read all about it in the front page of the newspapers, especially recently. And so this actually definitely dates myself, but we were only the second lab at Hopkins to successfully generate a CRISPR mouse line. And we used CRISPR to insert this V5 tag into the C terminus, into the end of the M-wave protein. So the reason we had to do this is, despite my first year in lab being dedicated pretty much entirely to buying, testing, even making a whole slew of antibodies, we were basically unable, I was unable to get anything to actually work and stain for this endogenous protein. So instead, we cut our losses and started to make a V5 fused protein. And now we could use the commercially available V5 antibody to confirm expression of M-wave in mouse tissue. And here you can kind of see an example of this wave V5 expression in the core of the SCN in mouse tissue. And then if you compare it here to, this is the mRNA as visualized with an in situ hybridization, and we can see that they correlate quite nicely. So we are getting good representative expression of our wave gene. Our second mouse, though, is more immediately interesting. Since the lab had already done these big, forward genetic screens looking for 5 mutants, we could skip all that and jump ahead directly into reverse genetics. So for reverse genetics, we again use CRISPR-Cas9, and this time we introduced a stop codon as well as a frame-shifting insertion into exon 5 of the wave locus. As you can see here, when we use qPCR to measure levels of wave mRNA, we can see that they're dropped by about 70–80% in the homozygous mutant. And the same is true when we look at in situ expression levels in the SCN. And that's because nonsense-mediated decay is chewing up this message here with the stop codon and all the frame shifting. But because this stop here is in frame, and this generates a whole number of stops as well, we believe that this does generate 100% knockout of the wave protein. So armed with this null allele, and with the information that we had gotten from the flies in the last story, we wanted to know what role does weight play in the regulation of sleep in mice. Okay, so sleep in flies is measured behavior by consolidated inactivity. Sleep in both mice and humans, however, is measured electroencephalographically, that works, which is called EEG. This technique measures the coherent activation, the coherent activity of many thousands of cortical neurons, either through the skull, like in humans here, or a lot more invasively in the mice, where we actually drop our probes through the skull and they rest on the cortex. This technique was developed a long time ago, turn of the 20th century, but by about the 1950s, scientists were regularly characterizing main stages of sleep and wakefulness with their characteristic waveform genres. For example, wakefulness, seen here, the EEG, has low amplitude, mixed frequency signal, and is often coupled with higher muscle activity, as measured on the EMG. In contrast, non-REM sleep, seen here, is marked by higher amplitude and low frequency waves. These are the slow waves, the delta waves that people talk about. REM sleep, the dreaming stage, is also called paradoxical sleep. And that's because the EEG waveform here actually looks a lot more like wakefulness. You can really only distinguish it based on the entire lack of muscle tone seen here. So, we took our wake mutants and I implanted these EEG recording head mounts into a cohort of homozygous knockouts and their wild-type litter-rate controls. And just like in flies, we wanted to look at their sleep over 24 hours. Okay, so here I've split each of the sleep stages into 3 different plots. Wake up top, non-REM here, REM here. On the y-axis is the percent of each 2-hour bin that the animal spends in each stage. And it's over 24 hours during the day and at night. Based on the fly phenotype, we were kind of surprised and kind of disappointed that the phenotype here is actually quite mild. There's a slight increase in wake at night in our mutants, and that matches with a slight decrease in sleep at night in our mutants. We also see a mild decrease in REM during the white phase. In addition, our mutants also have a harder time staying asleep for a long period of time, so it's also some increasing fragmentation and shorter sleep bouts in the mutants. But overall, the phenotype is actually quite mild. And this is, again, surprising and a bit of a letdown compared to what we saw in flies. But then we remember, of course, that mice are nocturnal, right? Mice aren't, both the mutants and the wild type aren't really doing all that much sleep at night, which is where the wake protein is actually active. What they are doing at night is running around. And I hope people can appreciate in this video that I haven't tagged the mutant because I hope it's obvious which one in there is hyperactive. So all night long, the mutants are going to run around the cage, they're bouncing off the walls, they circle around their friends, they're checking everything out. And this hyperactivity ranges quite a bit from mild to extreme. And in about 20 to 25% of our mutants, we found that they were quite content to spend many hours over the course of the night just running and driving themselves in circles. In order to measure this hyperactivity, we projected a mesh of infrared beams across the home cage of the mouse, and then we could track the mouse's activity as it would break these beams, and we record that as an activity measure. Using this system, we plot the total number of beam breaks over 24 hours, and then we can see things like, in this case, the nocturnal activity of the wild-type mice. They don't move around as much during the white phase when they should be sleeping, and at night they get up and they're running about the cage doing mouse things. The awake knockout mouse follow the same pattern, but they are extremely hyperactive at night. In fact, this is the same wild-type trace that I just showed you on that previous slide. But the y-axis is so stretched to actually include this hyperactivity that the whole thing gets squashed down. So when I say these mice run around at night, they really are up there and moving about for much of the night. After seeing this really dramatic hyperactivity, I wanted to go back and look at the organization of their wakefulness. And for this, I went back and looked at their the EEG traces that I collected before. So at the top here, this plot is called the hypnogram. And what this does is this shows the arrangement of behavioral states in the mouse. You can see that they are filled with lots of non-REM and REM states during the white stage when they should be sleeping. And then there are long bouts of wakefulness here at night, punctuated by a couple of naps every night. At the bottom, this is called a spectrogram. To generate this trace, we use a Fourier transform of the actual EEG waveform, and we break it down into constitutive parts and express it over a full 24-hour bit, just like above. And here, what I want to point out is, again, you can see lots of this low frequency, high power stuff. That's these red bars here, and that's sleep happening during the day. And at night, when the animal's up, you see more mixed frequency, lower frequency stuff all across the band. And again, those naps there at night. Okay. The wake knockout mice, however, have these ultra long bouts of wakefulness. Many hours a night. You can see it quantified over here. We're talking about 6, 7, 8, 9 hours of mice. I have more data that I can even add to this as well. We never see these kinds of long stretches in the wild. They punctuate their activity with bouts of naps here and there. In addition to this, so even though the total amount of sleep didn't change, as I just showed you, the organization really did. And while these animals are awake at night, their wakefulness doesn't look normal either. Instead of the mixed sort of lower amplitude frequency here, it looks like the mice are getting stuck on one frequency, and then the whole night they are just driving single frequency. If we saw like a little blip of this in a wild type, that would generally fall under exploratory behavior. But I don't think these mice are exploring for 10 hours straight. I think this is probably reflective of some of their stereotypies or their hyperarousal like this. Okay, so altogether, these phenotypes suggested that the wake mutant phenotype is really a disruption, it's a heightening of generalized arousal in mammals. So for mammals, the way we measured generalized arousal, or the way we assess disruptions to generalized arousal, is with 3 main criteria. The first thing is we look at the sleep-wake activity, which again, as we just mentioned, is definitely disrupted in our mutants with these long wake bouts. The second generally is locomotor activity, and I hope that showing this one again shows that these mice also are clearly hyperlocomotive. The third way to measure arousal in mammals is stimulus responsiveness. How do they react to cues that are coming in from their environment? To test this last component, I did a bunch of different behavioral tests, but the one I'm going to talk about today is the acoustic startle. So, in the acoustic startle test, Each mouse is placed on a pressure-sensitive membrane in this sound chamber, and then when they least expect it, we hit with a high noise and see how high they jump. The more aroused the mouse is, the higher jump, the stronger it reacts to this stimulus when it comes in a surprising manner. So when I run this test on our knockouts versus our wild type, what we see is that the wake knockouts are hyper-responsive to the acoustic startle, and they're hyper-responsive specifically at night compared to their wild type liturgy controls. The other thing we notice is that while I run this test, I get a series of loud noise mix that I have to do a lot of data collecting all at once. And normal wild type mouse jump higher for the first bit than they do for the last one. That's not that surprising. Our mutants, however, seem to be as reactive to that last noise as they are to the first. And this suggests both that their arousal state is heightened, but also that they don't seem to be habituated to these types of environmental cues. So I hope this is pretty good evidence that knocking wake out in mutants causes them to be hyper aroused in sort of every definition of the term. But what is it that's actually happening at the physiological inside the brain to generate this behavior? So to address this question, we turn to electrophysiology. In brief, electrophysiology uses a very tiny glass needle here and attaches to the cell membrane, your cell of interest. We can then record the spiking potential, the electrical differences that happen in the cell as an action potential travels down the length of the neuron. So Chung Lu is a postdoc in the lab who came in and really set hard to work getting all these electrophysiological recordings done. And what he did is he recorded from wake-positive cells in the SCN, in that master circadian pacemaker, both in the day and at night, and in animals that had wake and animals that didn't have wake. And because the clock has rhythmic cycling, what we saw is that these neurons fired higher during the day and lower at night, and you can see this effect quantified here, higher during the day and lower at night. When we knocked wake out of these animals, they fired the same amount during the day, but at night, the reduction in firing is lost. Something is happening, something that the wake protein does is responsible for this diurnal rhythmicity in firing rate in these SCN neurons. So the question, of course, is how are these changes occurring? How is wake affecting the daily firing patterns in these cells? So based on our understanding of flies, we believe it, and we suspected at the time, for a number of reasons, that wake was causing an increase in GABA sensitivity in the cells it's expressed in. So to test this, Chong recorded again from SCN neurons, from wake-positive SCN neurons, and this time he puffed GABA, the inhibitory neurotransmitter, over the cells and recorded their reaction to this neurotransmitter. Compared to wild type, the wake knockout cells had a decrease in responsiveness to the GABA puff. You can see that quantified here. Basically, something was going on that made these cells react less to this neurotransmitter. In cell culture, we can see that when M-wake is co-expressed with these GABA-A receptors here, it causes an increase in GABA-A receptor expression. So, altogether, when these neurons lose this heightened expression of the GABA-A receptor, then it's not all that surprising that they're less reactive to the neurotransmitter itself. But this effect is really only occurring at night, as we saw. And so what it suggests is that it's really the circadian clock that's pulling the strings here. So to test that, we looked at another type of mouse. This one has its circadian clock genetically abolished. This is a BMO mutant for the circadian people in the crowd. And again, Chong was reporting GABA sensitivity in the SCN of these mice as well. And it looks like that, again, in these mice, even though they have a functional wake protein, their responsiveness to GABA is blunted in about the same direction as the wake proteins. And what this suggests is that the circadian clock and wake act along the same pathway. And in fact, that wake is the tool by which the clock can actually tune the GABA sensitivity, tune the daily rhythm of firing in the SCN. So as all this data comes together, we can begin to construct a model of M-wake that looks remarkably similar to what we saw in Drosophila. So flies and mice exhibit conserved homology of the wide awake and M-wake protein. In both animals, wake protein levels are higher at night, which increases inhibitory tone, quieting the cell. And when you knock out the protein, this means that flies can't get to sleep, and so they become insomniacs. And our mice can't regulate their arousal level, and so they become hyperactive at night. of course, circadian clock. And we just talked about how oscillations in this circadian clock can drive changes in rhythmic cell firing by affecting wake levels in these clock neurons. Over the past year and a half or so, especially really in the past year and a half, our focus has shifted a little bit from the wake protein specifically and into the network of neurons that actually express the wake protein. So this is the wake circuit, and we believe that this defines a novel and fundamentally important arousal circuit in these mice. In order to start looking at the wake circuit, though, we needed a different type of mouse. This time we used traditional methods because CRISPR can't get this big construct in yet, but we're hoping soon. And we introduced a TV tomato fluorescent protein as well as a Cre recombinase into the M-wake gene locus. So this means that any neuron that would be expressing M-wake is also going to be tagged with this red protein and will also express Cre recombinase. Having this mouse now pulls double duty for us. First, because the TV tomato is under this wake locus, we can see that any cell that would express wake or does express wake is tagged with this red protein. You can see that again here in the SCN, and we confirm this expression by looking again at the C2 and comparing also to the wake V5. And more importantly, the Cre recombinase gives us genetic access to wake positive neurons. So Cre acts by splicing out regions of DNA that are bound by these LOX P sites. And we can use this Cre-LOX recombination in a couple of different ways. So let's say, for example, we want to knock out a gene of interest in only our cells of interest. Well, this wake animal expresses Cre in these neurons, but not non-wake neurons. The Cre that splices out this fragment of DNA, and we get a non-functional gene in our cells of interest. The other thing we can do is actually activate certain tools with this Cre recombinase. And these tools, then instead of being turned off, are actually turned on only in our cells of interest. And I'll talk a lot more about those tools in a little bit. After we realized, though, that the wake story was more about arousal and about sleep timing, we wanted to go through it and really carefully characterize where this network is, right? Where does this circuit exist in the mammalian brain? So, take a quick step back. In the field of neurophysiology, there are a collection of ascending arousal systems, which all interact with each other, interact with other parts of the brain to drive wakefulness, to drive arousal in mammals. A lot of us here are going to be familiar with some of these. I won't go over them in detail. But for example, there's norepinephrine cells in the locus coeruleus. This is the only source of noradrenaline in the brain. The VLPAG and the dorsal raphe have serotonin cells and have dopamine cells, which fire more during wakefulness or involved in reward salience. In the lateral hypothalamus, there are orexinergic cells, and when you lose orexin cells, oftentimes you have narcolepsy, as seen adorably in little dachshunds that fall over. And in the lateral hypothalamus, there are also histaminergic cells, histamine-expressing cells. And these cells are the reason why when you take an antihistamine, like Benadryl, you feel sleepy. It's blocking these arousal-promoting cells. And all of these cells are then linked to sleep by this region Vilpo, one of the few sleep-active areas in the brain, and it sends inhibitory tone to quiet all of these areas. As we started looking through the whole mouse brain, and working with, and I worked with Annette Wang on this, who was really a very, very talented math teacher who did a great job on this characterization. She and I were really struck by the pattern of wake-expressing cells that was in just about all of those areas that I just talked about. From the basal forebrain, through the lateral hypothalamus, the VLPAG, and the dorsal raphe, even as far back as the locus coeruleus, we saw these wake neurons everywhere. So we thought, okay, well, maybe these wake cells are also involved in the neurotransmitters. But it turns out that it's not co-expressed in the same cells as any of those neurotransmitters I just mentioned. So it's interesting that there's a population the population that expresses wake does not also express the neuromodulators and neurotransmitters. You can see that here with acetylcholine in the basal forebrain. Same thing is true with histamine here. Serotonin cells are not the same as wake cells seen here. And the same is true back in the locus coeruleus. So, if our M-wake neurons are intermingling with all of these neuromodulators, but they're not the same population, then what are the M-wake cells, right? What defines them? To do this, I use fluorescent in situ hybridization because it turns out it's really, really hard to stain for some of these neurotransmitters. And so using this, what I found was that most of the cells that express wake also express glutamate, glutamate. And this is really not all that surprising because most of the cells in the brain are glutamatergic, and because we know that wake activation drives wakefulness, and therefore it makes sense that an activating neurotransmitter would be expressed. But we also saw that some wake cells in certain specific areas, like the SCN and other regions of the hypothalamus, did express GABA. They were inhibitory as well. And this suggests that the network is involved in a couple of different pathways, involved in regulating different attributes of arousal. And I'm currently working with Thomas Kidd in the Blackshaw lab to do some single cell sequencing and look at more carefully exactly what the cell population of these wake neurons are. Huge thanks to him, he did a massive amount of work to actually get single cell sequencing data ready for this talk. But basically the data just confirms what we saw here so far, and we have to dive in a little further. So hopefully we'll have more information about the specific population of these wake neurons in different parts of the brain in the coming days and in the coming weeks. Okay, so to try and understand the role of wake in all of these different arousal pathways, we first looked at one specific region, the dorsomedial hypothalamus, the DMH. The DMH has been previously implicated in coordinating clock-driven arousal, and there's really a big body of literature that ties activity in the DMH to a number of arousal behaviors and arousal pathways. And importantly for us, the DMH here sits at the nexus of the core circadian clock and these arousal pathways. The DMH receives direct input from that core clock, the SCN, and then from there it sends activating and arousal cues out to many downstream pathways. If, as we suspect, the wake cells in the DMH are responsible for this arousal promoting activity, then by manipulating wake in only the DMH, we should expect to see a similar hyperactivity phenotype. Okay, so to look at wake in just the DMH, we had to make yet another mess. This time it's a conditional knockout in which we surrounded an important exon in our amyloid locus by these LOXP sites. We can then introduce pre-recombinants inside the virus by stereotaxically injecting directly into the DMH of these mice. And as you can see here, this is just a representative image. The CRE that's bound to a GFP is expressed in all these cells, and in these cells that it's expressed, it knocks out wake protein. I can then take these animals that had their wake knocked out in just the DMH and run them in the beam break rigs to look at their activity. And what I did is I compared their activity before the injection to their activity after the injection, after the wake was knocked out. What we saw is that wake levels, when they're knocked out in the dirt, mice that have wake knocked out in only the DMH also experience hyperactivity. And we compared that to a different set of mice when I inject with them with the same virus, except this time it's a sham virus. The CRE doesn't actually work. We see no change in their activity levels. So clearly, the knocking out of wake is what's driving this. Okay, so if losing weight is driving this activity, again, what's happening at the physiological level? This time, John reported from wake-positive neuron from the DMH. And the DMH is different from the SCN, right? The SCN is the core circadian clock, but the DMH is an output center. The DMH cycles in phase with the activity of the animal. So in this case, during the day, when the mouse is mostly sleeping, firing in the DMH is low, but at night, the firing rate in the DMH increases, so it's a nocturnal mouse. That same pattern is true even when we knock out the wake protein, except at night, this time, those cells fire rapidly, so it's an increase in activity in these wake neurons in the DMH. So this is actually a really nice connection. We like this a lot. It shows that wake cell firing increase and there's an increase in activity, but ultimately it's still circumstantial, right? What we really need to show is that if we manipulate those wake cells directly, manipulate the wake circuit directly, can we then affect behavior without going through the protein at all? So to affect just the wake neurons, we turn to a class of tools called chemogenetics. These are also called DREADs, designer receptors exclusively activated by designer drugs. I would say that. In this case, the designer receptor here is a DREAD GQ. This is a special G protein coupling receptor that when we give the mice its designer drug, CNO, it binds to the receptor and causes rapid activation, rapid firing of the cells in which the GQ is expressed. We can control which cell is expressed by using our wake mouse. So, we injected our DREAD virus into the DMH of wake-free mice, and again, gave them CNO and then tested their activity. And what we can see is that when we drive activity in just those DMH cells, or drive activity in just those neurons, we see an increase in locomotor activity in the mice as well. So now this chemogenetic experiment shows us that it's the activity of the wake neurons in the DMH, divorced from wake itself, that are actually driving behavior. And this direct connection here is what allows us to define a new arousal circuit. But this new arousal circuit is bigger than just the DMH, right? As I showed before, wake is expressed in and around most of these arousal areas, and sort of poorly represented by my red dots here. So, and all of these areas work in concert to drive the experience of arousal in a mouse. And what we thought would be that the influence of wake cells reaches beyond just where its cell bodies are located. So, to look at the region of wake cells, we use another virus, this time containing a GFP protein. And this is something that myself and Sam Lee and other really talented postdocs in the lab worked on. We injected this GFP virus into just the wake cells in the DMH of these mice. And we looked for the spread of their projections, right? So as the GFP fills these projections, we can see where it sends its reaches throughout the brain. And we look at a sagittal section of our GFP containing virus that was injected here. I don't know if you guys can see it as well. I apologize. I hope this image is as striking for you as it is for me, but it's probably not. No. Okay. Anyway, we injected here, and we were actually quite surprised to see that the projections, the processes from these cells reached all the way through the brain. You can't see it here, but there are a number of projections that go back down to the pons in the midbrain, all the way back to the locus coeruleus and the VL pack that I talked about. They go back down forward to the basal forebrain, and a number of these projections reach all the way through here, up and around, and then they spread up to actually innervate the cortex. And so now our idea of what wake is doing is a little bit more complete. We can see that the cell bodies are dispersed in all these areas, but it's also that these cells send big projections through both to each other, to these other areas, as well as the rest of the brain. In order to fit this expanded wake circuit into what we know about the physiology of arousal, I'm going to take one pretty big step back and talk about what the general framework for arousal is. For the past many years, psychologists have modeled human arousal on sort of ascending levels, an ascending period, where each level is required for the one above it, and they also talk back and forth to one there. At the bottom is basal arousal, right? This is the whatever level of arousal distinguishes our brain from somebody in a coma, distinguishes our brain from just a ball of neurons in the same shape. From this basal arousal comes cognitive arousal. Cognitive arousal is the type of arousal that we're all more sort of familiar with. This is the level at which we can distinguish sleep from wakefulness. This is the level at which we can be hyper-excited and ready and activated. Above cognitive arousal is attention. This is the directable focus that we can apply to day-to-day problems in our and other animals' lives. Neurophysiologically, there's a rich body of literature that tries to assign neurological substrates for each of these levels. So for example, I really am oversimplifying, sorry. So for example, attention is thought to be directed by the cohesive activity of certain components and certain cells in the cortex here. The neuromodulators here are thought to drive and change the accessibility, the onset of sleep and wake, as we see in cognitive arousal, as well as a number of other cognitive arousal measures, like sensory responsiveness, learning and memory, etc. But for many years, much of the focus in the neuroscience field is really on these neuromodulators. How much does norepinephrine affect sleep? Right? How much does activating histamine drive arousal? But we know now that these neuromodulators, they don't tell the whole story. Their individual effect on behavior is limited, and if we lose one, we don't totally change the behavior. Instead, there has to be some other physiological substrate that's actually responsible for the type of arousal that the rest of these are built on. What we know is that it's likely that this fundamental circuit would have a couple key attributes, right? It's likely that it would be glutamatergic. It's likely that it would connect and be located around many of the other arousal centers I've been talking about. And we know that its activity would likely be tied directly to behavioral activity in the animal. It looks like the wake circuit actually fit that description to a T. And so we began to suspect that wake is acting as the fundamental arousal circuit in mammals. To test this hypothesis, we turned back to chemogenetics, and we had to do two key experiments. In the first, we express the activating dread GQ in all of the unwake neurons, not just the DMH, but everywhere that there's a wake cell is now expressing this activating this activating dread, and we get activated with CNO. We also use a similar tool called dread GI, and this is an inhibitory dread. In this case, we can express this dread GI in all the wake cells, and then when we give them CNO, we actually silence the entire wake circuit simultaneously at once, and then we're going to look for the behavioral outputs of these animals. So, in the first test, based on everything we've seen so far, it's really not all that surprising that when we put in the activating dread GQ and then give them CNO to activate the wake circuit, the animals had sustained wakefulness. They were pretty much awake for 6-7 hours after giving the injection and were effectively not asleep at all during that same time, compared to the same group of animals that were injected with vehicle, with a control at a different time. If we break down the frequencies of the raw EEG signal from when we treat these animals, we can see that this period in the vehicle animal, a couple hours after injection, is filled with sleep, right? So it's nighttime, the animal should be asleep. We give the vehicle injection, they go right back to sleep. On the other hand, when we give them CNO, the couple hours after injection are most certainly not sleep. Not only are they wakefulness, we can see behavior, it's also a really intense type of wakefulness that extends for 5-6 hours, and then when the lights go off and the animal should be waking up, it's so tired that it has to go back to sleep then and engage in some recovery sleep, some rebound sleep. So I guess the reality is this isn't all that surprising. If we activate all the wake cells, we drive wakefulness. This doesn't really distinguish wake from any one of the other arousal pathways because this happens to a lot of them. If we use the dread GQ to activate all of the norepinephrine cells in the LC, we can also keep animals awake and prevent sleep. Some humans take cocaine to activate their dopaminergic system, and they also are not known for sleeping for long hours at a time. So, although we can drive wakefulness, basal arousal isn't dependent on just sort of driving an individual component of wakefulness. It's a required level that everything else builds on. To address that question, we had to inhibit the M-wake circuit all at once. And the spoiler alert is that inhibiting the wake circuit simultaneously had a really dramatic effect on general arousal in the mouse. I want to say that before I show the video so you can get a chance to pay attention. In these two mice, they're both dread GI. On the left, I injected with vehicle. On the right, with CNO. And this is about a minute after I injected. They're both up, they're parked around, they're sniffing, trying to figure out what's going on. They have no problem hanging out on top of their perch and seeing what's going on there. Fast forward 30 minutes. This is the same mouse injected with vehicle, and this one with CNO. The vehicle mouse looks fine. It's not having much of an issue at all. The CNO mouse, however, is clearly not in its normal state of mind. It has its body posture is slack, its muscle control is reduced. It keeps falling off the perch as it becomes, sort of essentially non-responsive, right? This mouse can't keep itself up. Here's another example of what that global inhibition of wake circuit looked like. It results in basically a silencing of responsiveness. Here you can see two new mice. On the left, this guy's treated with only vehicle. On the right, with a relatively low dose of CNO. And you can see that the vehicle-treated mouse really does not want to be poked by my technologically advanced stimulus stick. But if I do the same experiment in the CNO, he's either unwilling or unable to respond to this cue. I mean, it's a pretty significant cue as I poke him with this. What's kind of interesting though is he does maintain his writing reflex and other really basic physiological functions are maintained. Breathing and thermal regulation. That's all still going on, even though the responsiveness is totally lost. So I think after seeing those videos, it's not really surprising that when we gave those dread GI mice CNO and put them into a locomotor assay, they pretty much dropped to zero. Shocking. So to understand what's happening neurologically, we had to look at the EEG signal. And when we look at EEG signal, I think you can appreciate here that in the CNO treated mice, the cortical activity dropped dramatically. It's very low amplitude and low frequency compared to, this is the same mouse, just on a different day when I treated with a vehicle control. If we convert this EEG signal to a spectrogram, we can see here the complete loss of cortical activation. When we silence the wake circuit, this period here is not really sleep. It's far more comparable to deep anesthesia or some types of coma. And what's striking is that even after going to pretty close to cortical silence, After a couple hours, when the CNO starts to wear off, the animal can recover back into its normal periods of sleep and back to baseline. To my knowledge, this is the only genetically defined circuit where inhibition is going to cause this kind of reversible cortical silence broadly in the animals. So we saw that if we inhibit all of M-wave, our mouse doesn't just fall asleep, he actually goes, if we block the signal, he goes way deeper like anesthesia, deeper like coma. And what this indicates is that at least some activity is required in the wake circuit for any of these higher levels of arousal to occur. And we think that means that this makes this wake circuit the source, the basal source of arousal in mammals. If we're going to argue that that's true, the other component of this that we need to show is that this basal circuit is interacting in a bidirectional way with the neuromodulators. And so, first, does it interact with other neuromodulators? I already showed you that physically, wake neurons send their projections throughout the brain, and they go and they intermix with a lot of these other arousal centers. Sorry, you can't see this clearly, but trust me, really, they go everywhere. It's very impressive. But do the other neuromodulator neurons connect back into the wake circuit? To do this, we use rabies bacteria sequencers, which when we inject into a wake positive neuron, and any neurons that then form a monosynaptic connection with this, uptake a blue fluorescent protein, and we can then stain these cells to determine their cell identity and see what's actually connected. And it seems that many of the classically arousal-promoting pathways directly connect to the M-wave circuit. We can see that histamine cells send direct connections. Orexin cells, all in the hypothalamus, also directly connect. As far away as the locus coeruleus, norepinephrine, noradrenergic cells, not all of them, but some send their projections all the way in and connect directly to the wake circuit. Even acetylcholinergic cells in the basal forebrain, they also have a couple cells that come and form monosynaptic connections. So, we think that all of these arousal centers are now connected through the wake circuit. What's the functional reality of that? Right? What does that actually do to the wake circuit? There are a lot of really complicated ways that we can address that question, but we started with a simpler experiment. In this one, Chong is recording from wake cells in the DMH, and then he's going to spritz the cells with one of these arousal-promoting neuromodulators, epinephrine, orexin, or acetylcholine, and record their reaction to that exposure. And so, as you can see here, whether it's norepinephrine or orexin or acetylcholine, when Chong would actually spray the neuron with the neuromodulator, there was a dramatic increase in firing rate compared to before. You can see that that's quantified for all three of these after as well. But all three of them stimulated firing in the wake circuit. And so, again, we consider our hypothesis to be true. This isn't all that surprising. An increase in the arousal-promoting neurotransmitter causes an increase in the activity of a fundamental arousal circuit. Makes sense. But is the other direction true as well? Does activating the wake cell activate other arousal networks? For this experiment, we did have to get a little fancier. Here we're using a tool called optogenetics. Optogenetics takes advantage of a blue-light sensitive ion channel called channel rhodopsin. We can express channel rhodopsin in just M-wave positive neurons using our wake 3. And then when we shine a blue light on those cells, it causes a dramatic increase in firing in those cells specifically. In this case, we put our opto expression into wake neurons from the DMH. And then Chung would record from norepinephrine cells in the LC. So then when he was turning the light on, what he was doing was activating the wake cell and seeing what would happen to this norepinephrine cell. And in this direction, we see that as soon as we turn the blue light on and cause this cell to fire, we saw an immediate firing rate change and an immediate response in the downstream norepinephrine cell. What this shows is that there's a functional connectivity between the wake positive neurons and the locus coeruleus norepinephrine cells that are really very well established in arousal literature and arousal generation. So we put all this together. Wake positive neurons are expressed throughout many of the arousal centers in much of the mouse brain, and they send far-reaching projections both to each other throughout the rest of the brain and to all of these other established areas. The network also has bidirectional functional connections with many of the arousal centers that we see in this brain that are involved, that are previously shown to be important in regulating arousal. We know if we activate these cells, the mouse stays wide awake. But really more importantly, if we silence these cells, the mouse enters a coma-like, an anesthesia-like stage below the level. So the activity here is required for the animal to rouse itself. And so, because I'm a little bit low on time, I'm not going to go through the whole summary, but I just want to say that we started with the circadian clock and showed how the clock tunes the arousal and tunes the gain on arousal-promoting cells by inhibiting or by increasing inhibition or decreasing inhibition in these arousal-promoting cells. But the activity of these cells themselves are really what's important. The ones that express weight are involved in generally regulating the basal function, the basal arousal level of an animal, regardless of weight protein. We knock it out, we see an increase in basal level because there's less inhibition. But if we just activate the cell, we see another hard driving activation of these neurons in general. Okay. So I said if I was running over, I wasn't going to go through my future directions, so I don't have time. But they're really interesting, and you should ask me about them. We are using weight as a model for a couple different disease pathologies, and we have some really, really cool experiments that are going to be starting basically right away. So you can ask me about those. I'll say this all the time. I do however want to give some very serious thanks to everybody on this list. I'm not going to be able to go through and really get everybody, but especially obviously especially Mark and Chung did a huge amount of the electrophysiological work and came ready and hit the ground running. Sang-Soo also did a great deal of the projection mapping and trying to understand where he's projected to. Annette was awesome to have as a master student for about a year and a half, and she really went from sort of just getting into the mouse brain field to becoming a really stellar mouse physiologist. So it's impressive. Everybody else who Yvonne, who did a ton of mouse work and came on and joined Ian, who answered endless questions and continues to answer endless questions. Thank you. And everybody else allowed really did a good job. I can't thank them all enough. The whole Black Shot Lab has been really helpful both in terms of ideas and giving us access to new techniques and new tools there from the beginning. And Thomas and I are working pretty closely right now, so thank you very much. The behavioral core, the Cohen Lab, Stammer has been really, really helpful this whole time. One of the especially thank my thesis committee. As you guys can see, I'm running over now, but you should have seen it when it was the thesis committee. And they tough it out to several 3-hour meetings. So thank you guys very much for coming. I appreciate that the party really took shape on the backs of this. And then I would say, thank the Hopkins Human Genetics program, the program that I'm in, David and Sandy and Kirby, thank you for coming. I really appreciate all that you guys have given me over the years, and the whole lab here as well. With that, I will take any questions.
Is there anything known about the human ortholog for this gene?
Morgan and I were talking about this. It has to be the first question anybody's going to ask. The short answer is because it's so similar from flies to mice, and the protein itself is really similar between mice and humans, we suspect that the human protein is probably doing about the same thing that it's doing in mice, except it's promoting sleep because it's acting at the nighttime when humans get to sleep. As far as disease models, I've been looking in as many of the repositories as I can to look and see whether there's any diseases known from mutations in M-Wave or anything like that. And to our knowledge, nothing really jumps out. There's a couple of hints from some GWAS studies, like addiction, as I mentioned before. But there doesn't seem to be anything. There's a little bit of selective pressure against mutations there. So it's probably important. But I don't think it's known exactly what it's doing in humans.
So what are your thoughts about the wing cortical targets and the nerve circuitry that might be?
So there is a stretch of weight neurons in layer 2, maybe layer 3 of the cortex that we don't know what it's doing. Is the short answer. One, we're not cortex people, but I think it's been shown that it is people are interested in that area because it gives genetic access to a population of neurons that are currently understudied. But what that means is that we don't really know what those neurons are doing because they're understudied. One thing that I'm pretty interested in doing going forward is kind of breaking the circuit down into component parts and seeing which areas are responsible for which outputs of those outputs. Cool. Okay. just want to give a couple of really quick acknowledgements to people who have supported me far above and beyond my entire 7 years here. And so first, there's no way I can thank everybody. So please know if I met you at some point, thank you. I'm going to try and keep this. I'm not going to cry. But I just want to thank a couple of people who are here who've been a big part of this. Obviously, first, thank you very much to Mark. Mark's really an outstanding person, an outstanding scientist, and for me, it's been a really outstanding mentor. I'm never really going to be able to properly convey, what our relationship was and how much I think it's had a positive impact on me. But I hope that you know that I'm sincerely appreciative, and grateful for everything you've done in the past 6 years. So thank you very much. When I first rotated in the lab, all the way back, if you're going all the way back, I will too. When I first rotated in the lab, I remember, I think it was like our second meeting or something. We were in there, we were talking about the work that was getting done and not getting done, and Mark was like, listen, Ben, I think you can be here, but right now you're here. So I need you to accelerate that move up here. That was my rotation, my first time ever touching flies. And I remember going home and being like, wow, that kind of is rough to hear, but it's very, very motivating in the sense that he thinks I have this potential, and he's probably right. I'm not doing enough at this point to accelerate my growth. And I think for many of the first meetings that we had, that was a really common theme. You need to be up here, right now you're here. And I think that was very helpful in getting me to work harder, to pay more attention, to be single-minded in my focus and single-minded in my pursuit. So thank you very much for that. And I know that Mark, we all work pretty hard in that lab, but I think what's nice about working hard in that lab is it shows you how much you can do. If you have someone lighting a flame, you're like, hey, let's go, let's do this. We know this is capable, we know we're capable of this, so let's get it done. I think that's a really good recipe for success. So thank you for that. And the other thing I really wanted to point out is one thing that Mark does that I think is really valuable in terms of leadership, in terms of mentorship, is he is always an open book. That means every decision that he makes, every time we have a meeting, every time we decide to do an experiment or decide not to do an experiment, every time we put a sentence in or take a sentence out of a grant, Mark explains his reasoning, his thinking about why that's happening. We don't always agree on the why, but I appreciate the fact that by explaining his logic, I can incorporate that logic and that way of thinking into my own growth as a scientist and to a leader going forward. And I also really appreciate that Mark is very big about the work-life balance, and even though when we're at work, we're working, there's always these hints of a life outside. When I first joined the lab, there was a line on Mark's profile that said Mark was a DJ in college, and I almost couldn't believe it. I couldn't picture that for years. It haunted me. There were these little clues here and there. The music that was playing, I had these really obscure band T-shirts, and Mark, all we knew was what was going on with that. But it wasn't until recently that we found some evidence that and I'll bet he was very, very damn good at it. So thank you very much for everything. I really am grateful. I also really want to make the Woo Lab. This is not even when I started. This is like a year and a half after I started. We were pretty small. This is for Chi-Lee's that departed postdoc, her baby shower. And in the time from that point to get into our conference room, now we've had to move all of our lab get-togethers either outside or to somebody's home. Because we've really grown as a lab as a community. I had to pick two images so I could try and get everybody in there. And so I think it's I think it's a testament both to Mark and to the lab. The quality of work that's coming out of there is really very, very impressive. There's a big collection of expertise that grows every day, and I'm always amazed by the fact that I can go in there with questions about almost anything. It's somebody in the lab that's thought about it or is willing to put down what they're doing and help me figure it out. So that's really appreciative. And I feel very fortunate that the body of work that's happening in the lab right now is going to be really powerful in the future. And I think all of that is going to be reflected glory on myself. So one day I'll get a tag out of the Woo Lab. There's going to be a huge long list of papers that reflect on me anyway. So thank you guys. That's work that you've done. Thank you very much for it. And again, I would thank everybody here if I could, but I just don't have the time to do everyone. So please know that I'm grateful. I'll corner you and gladly we can talk about it. I also really need to thank my family who supported me from day one, truly day one. They've always been there. Both of my parents are doctors, and yet they managed to not push me into the medical field, sometimes harder than others. And they supported my interest in science, my interest in everything. They were constantly reinforcing the idea of ask questions, go figure out your own answers. If you want to know this, I can tell you the answer, or I can tell you how to look it up. And I would go and do this research. They would spend any amount of money, any amount of time getting me the education, the fun, the education that I needed, taking me to the library late at night. I still go home, and probably quite literally a metric ton of books sitting on the ground from when I was a kid. There's all there in a big library. So I'm very appreciative of that upbringing. And then also as an adult, my mom continued to push me to try new things. She had to hear all kinds of complaints when I was struggling in the lab, but then she also, said, push through it, it's worth it. We did a triathlon, that's sort of a really physical representation of pushing through it, it's worth it. It was not worth it. And so thank you very, very much for that. I appreciate it. I couldn't have done this without that. And my dad as well, he also, as long as I can remember, we used to drive Colorado drives in New York, and instead of just listening to the radio like a normal person, he would take these great lectures, these huge these huge tape decks that had books of hundreds of tapes, and plug it in and be like, oh yeah, I know I'm a doctor, but now I'm going to listen to 300 hours on quantum string theory, just because I'm generally interested. And I would complain bitterly, but at the end of the day, this is all, this is ideas and their concepts sort of osmosis their way in. So I'm appreciative of that, and he's also been very encouraging along in here, even when it's been tough. Other family is my brother, who made here. Morgan, thank you for coming. It's me. I have a younger sister that's forming a pancake with my much younger sister as well. She could not be here. She's too busy being angsty in high school. But both of them do make me feel like an expert, right? Which is nice. Maury and I have a chance to go out and do fun things, and he's doing a bunch of pictures of friends later. But he also supports the fact that this is a hard job. He's in grad school now, so clearly haven't warned him off it enough. My younger sister really makes me feel like an expert because she's in high school, and so when she has a problem in one of her science classes, she'll call me, and then when I say look it up, she goes, ah. So it's nice to have that support from her. In addition to my current family, I have a future family. Just go ahead. Thank you for coming all the way from our apartment. I met her through the program, and in the time since she has somehow managed to sit through probably the toughest time for this PhD, and the times I've known her from the times that I've been in the lab for long hours, going to see my mistress, away all the time. She really dealt with it with well and painful, and encouraged me to do the stuff that I had to do, and I'm very appreciative for that. And I'm also really appreciative of the fact that when I need a break, when I need to step away from the lab, she's really always there for that as well. We go hiking. I don't know where this is. We go hiking. We go to the Renaissance Festival, which is also a fun trip. I almost proposed here. I'm probably pretty bad at it. I didn't do that. And sometimes when we need to get away and just be silly, she goes and stands near huge giant floats. So I'm very, very appreciative of everything you do, and I'm excited to start that. And then there's also the huge friends that really were very influential in my time in Baltimore. I moved here from college in Boston and came in not knowing anybody, and I feel like I was really adopted into a big circle of friends. I can't thank everybody here specifically, but you know who you are. Thank you very much. Thank you for coming. I appreciate you being there for all this, whether we're at a wedding or we're going whitewater rafting in West Virginia. And I also particularly want to thank Joel Benjamin and Anna Mose, who really took me on as their adopted kid, even though I was only a couple years behind them, and being a place to sleep on their couch on Sundays and learn from them and learn from Joel what it was like to be a PhD student. And so I'm very appreciative of all that he's taught me. We also have a wide array of sports, but I'm not going to spend much time on it. I'm very, very thankful for the people on these sports teams. We did a lot of winning. Recently we've been losing, but we have to change that. I've been busy. Okay. I also really, really, really want to thank my crew of friends. These are people I knew from college and beyond college, and we still have a really tight circle. You don't get to see them as often, but when we do, it's like, time's passed off a number of years. Danielle and Josh are both here. Danielle drove an hour and a half to come see this, and Josh flew from Cincinnati to come see this talk. Neither of them are biologists, so I'm very happy for the fact that you guys can come and do this. And we, as a circle, have a lot of friends. There's people here. We support each other as people have good parts in their lives and hard parts. It means the world to me that we can have this, and that we can get away sometimes and go build geodesic films and big festivals. It's really meaningful, and I think it's important to my sanity and to everybody's sanity to have kind of like a nice tight circle. So thank you very much, guys.
Final announcement, you're all invited to the after party. It's at Rango's 271 immediately following. Thank you. And there's food and champagne.
Yeah. We got
How's everything? How do you guys understand that? Bits and pieces, I realize one of the things that always takes me a lot of reading back and forth with biology and genetics is how inhibition works and like what's being inhibited, like it's
be very complicated.
yes, like okay, so an inhibitor gene promotes activity.
An inhibitor gene promotes
So I know what
activity.
an inhibitor is.
Right,
It
I'm
depends
saying
on
the
what
loops,
gene-
the loops here, you have to get very involved in this through
Right,
every day.
right.
Because the promoters versus inhibitors gets me backward.
Because generic inhibition would be like causing a state of disarousal, whereas
Makes me feel very dyslexic.
yes,
That's what
oh
I'm
my
saying. I'm
God.
so far out of the science, it blows me away.
Yeah.
You know,
I just want to show
I
you.
did understand a little
I
bit.
just
So
want
you
to
got a dog.
show you the dog we got. Yeah.
Big dog, little dog.
It's about 65 pounds.
Oh, no, how old is it?
She's 3 years old.
You get a pound?
Rescue.
Yes, a pound. That's very nice. Housebroken? Was it an abused dog?
Not really sure. Her tail's docked, so which is nice because last round on the coffee table.
Well, the other thing is if it was an abused dog, once he hooks onto you, he'll never leave you alone. He'll be protective. It's just awesome.
Yeah, I don't know. We don't really know a whole lot about it. Its history. But
You know, that's
things,
great.
yeah, I think there's about a teeth. I think it's 3 in a month. Stealthy, she's kind of chunky, so we're going to lose another 5 pounds.
Letting the dog alone.
But yeah, she's a whole lot of love. So
That's neat.
yeah, I was going to show
So
her off.
since you're home all the time, you get to walk it.
Pretty much. I would say, to be fair, Meredith adopted a dog. I'm just taking part in this.
just
scared.
watch-
I told
watch
her.
it.
I told her I can't be financially responsible for any part of this. I'm happy to help. I can't afford this. It's
Get
fine.
up for a dog. Well, of course you can afford a dog. Yeah, no, you can afford a dog.
It's mostly pretty good.
People had asked me that before. My favorite part is when people are like, oh, we thought about looking at this. Actually, I have like 16 slides. Let me go look at all of the data. I haven't talked about it during the actual. I did it. That's what I was asking you. Well, I said I like questions like that, especially when I have future. I have a whole. There were 50 slides in that talk, and I have 130 slides. I have a lot of questions, a lot of.
So where is it going to go? Where is the big, where do you think?
All the week I'm supplementary pages. I'm going to have a 4-figure paper that has 17 supplementary pages.
No, that's not what I was asking. So where do you think this, in the, autism, the ADD, all the facts, where do you think?
I think, I mean, it should save it. Just like,
Very much basic science.
this is so good. I mean, mouse trace. Oh, alright. Well, I don't know. Where, I don't know. Should I try X?
I bet if you do that, it's just.
That's a color thing, I think. I'm gonna try Xing.
Yeah, I mean.
Ben. Okay, so it's different. He can play, but I don't want to watch it. Stand. No.
I was like, fucking with this.
Ben, you're still live streaming. We don't know.
Last for questions.
You're,
Is there not like a button that turns?
I wasn't sure if like you need to do anything to make sure you save it.
Oh.
I didn't want to touch anything.
I don't know. That's a good question. I should.